Assalamu alaikum wa barakatuh. Good afternoon, everyone. Thanks, Dr. Hani, Dr. Majid, for organizing this much needed educational symposium in order to learn how can we safely implement the switch of biological reference medicine, the blockbuster biological reference medicine, to the biosimilars at the Ministry of National Guard Health Affairs. And that's going to be a national guidance for the rest of the hospitals and the other education healthcare sectors, actually. And thanks, Dr. Hani, for excellent introduction. I don't have any disclosures. Thanks. I don't have any disclosures. And as you know, that after the loss of the patency of many range of blockbuster biologicals in the field of oncology and hematology has resulted in the development of new class of compounds called biosimilars, which are quite cost effective and were able to contain the global oncology expenditures. And these blockbusters, monoclonal antibodies, namely trastuzumab, bevacizumab, and rituximab, they had billions and billions of dollars of business around the globe. And now the international regulators such as EMA, US FDA have approved their biosimilars. There's some of them have 4 or 5 biosimilars of these biological reference medicine. And likewise, Saudi FDA has also approved the biosimilars of these 3 blockbusters monoclonal antibodies. And the Ministry of National Guard Health Affairs is the first healthcare sector who has adopted all these 3 biosimilars. Firstly, we approved trastuzumab biosimilar, but actually we didn't have it physically in-house. And recently, a couple of months ago, we have approved the biosimilars of bevacizumab and rituximab. So the purpose of this meeting, getting together, is to understand the perception of the biosimilars. So many physicians attending and pharmacy colleagues and nursing colleagues and other colleagues from the medical societies, they have been voicing their concerns about the biosimilars. They're like generic medications, something like poor quality medications. And then the industry had a significant influence, particularly on the physician and pharmacy side, pharmacy part, that you are going to use something which is of inferior quality. So we're trying to address these kind of concerns, which are voiced by medical societies, especially the switchability and extrapolation, these kind of concerns. We'll talk about pharmacovigilance. My colleague, Dr. Iman Yusuf, she will talk about that. And my colleagues, physician colleagues, they will talk about like how can we integrate them into the oncology practice, and they'll be presenting the landmark trials for that. And I'll present one of the landmark trials, just an example to show you many physicians and many pharmacy colleagues at several levels of discussion in the meeting, they have been saying, no, the biosimilar do not need any clinical trial for the approval. So we're gonna come up with this kind of, addressing these kind of concerns. And lastly, I'll talk about the formulary consideration of the biosimilars at the Ministry of National Guard Health Affairs, the journey that we have taken successfully, and the pharmacoeconomic aspects of the biosimilar addition to the formulary. That's what we're gonna share with you. First, let's try to understand the definition of the biosimilars. So biosimilar is defined as a highly similar copy of commercially available biological reference medicine whose patency has already been expired by international regulators. And this is highly similar, not identical. It's highly similar in terms of physical chemical characteristics, safety, efficacy, and immunogenicity. Let's try to understand the approval process for the biosimilar products. If you have questions in your minds or misconception about a biosimilar, so we can address some of those misconceptions by going through the development program of the biosimilars by the international regulators. So the manufacturer of the proposed biosimilar, it generates an array of data in a compatibility exercise, trying to demonstrate the similarity or the biosimilarity of the proposed biosimilar with its biological reference medicine. And this compatibility data is generally evaluated and carried out in a stepwise fashion, beginning with the analytical data, which is structural and functional characterization of the biosimilar using the state-of-the-art tool. And once it's found to be comparable to the biological reference medicine, then they move on to animal studies and PK/PD studies, and then they move on to the human studies. And they have to do at least one clinical trial in the most sensitive indication in order to demonstrate its compatibility and its similarity in all these stages. So a biosimilar has to demonstrate its biosimilarity or compatibility at each step. If it does not demonstrate its compatibility or similarity at any step, it will be failed. So international regulators will not approve it. And international regulators will approve the biological license application only if the biosimilar has demonstrated its compatibility and similarity at all the stages of the development. So these are some of the key differences in terms of development of the originator, which is the brand biologicals, and the biosimilar, which is the copy version of the biosimilars. So the purpose of developing the originator, for example, Maptera, for hematologists to understand, and Herceptin for the medical oncologists to understand. So they have to establish their own clinical efficacy and safety. The purpose of the development of the originator is to demonstrate and establish its own efficacy and safety data and immunogenicity data. And the most of the effort for that reason is actually carried out in the clinical part. So you need clinical trial for each and every single indication. The international regulator like US FDA, EMA will approve each indication based on the clinical trial carried out in each indication. That's not the case with the biosimilars. The biosimilar, the purpose of development of the biosimilar is not to establish their own clinical efficacy and safety, but to demonstrate its similarity with the originator. And for that reason, the most of the effort is actually carried out in the analytical part, trying to structurally and functionally characterize the biosimilar and prove that it's similar to the originator. And the least effort is carried out in the clinical trial. And that's what the originators industry is trying to come and tell physician and pharmacy and nurses and other medical society colleagues and healthcare workers colleagues that there is a minimum clinical trial. And for example, the extrapolation sort of issues that this particular drug was evaluated in early stage breast cancer, in HER2 positive breast cancer. How can you apply the data for metastatic breast cancer? So these are the concerns that we're going to address, that the purpose of development of the biosimilar program is not to go through each clinical trial, because the purpose is to have cost-effective molecules, cost-effective biologicals, to reduce the healthcare expenditures by at least 30 to 50%. For that reason, you do clinical trial only in the one of the most sensitive indications. Okay. I think there is some problem with this. Okay, now this slide differentiates between the generics and the biosimilars. So many times I heard in the P&T committee attending meetings with different physicians and other healthcare workers, oh, it's just like generic. Especially when we're talking about rituximab biosimilar. So some of the non-oncology physician colleagues are saying, no, this is not approved in XYZ indication. It was only evaluated in rheumatoid arthritis. So what about ITP? What about other indications? So basically, the generics are simple structure. You can create identical copies in comparison to the biosimilar, which are complex protein molecule. And it's impossible to maintain its molecular structure over time. And sometimes there is variation in between the different batches of these blockbuster monoclonal antibodies. And for the approval process we just went through, you need clinical trials for the biosimilar for your approval. You don't need clinical trial for the generic medication. You just need the PK/PD studies. And cost to develop and bring generics to the market is much more lower than that which is associated with the development of the biosimilar. These are the practical issues. First couple of issues, switchability and the extrapolation of the biosimilar across the indication of the biological reference medicine. When I use the word biological reference medicine, this means this is the original brand, which was first biological, which was approved by the international regulator, and biosimilar are just the copy version of that biological reference medicine. Pharmacovigilance issues will be talked about by my colleague, and we'll talk about the formulary consideration. So let's first try to understand and address the concern of switchability from the reference medicine to the biosimilar. Is there any concern? Is there any risk of switching? Before we answer this, let's try to ask another question. Is there any risk of switching between the molecularly dissimilar drug in comparison to the molecularly similar drugs, I think the same international non-proprietary names? On the left-hand side, on your left-hand side, you can see these are, I think hematologists are sitting here, my friends, Dr. Ahmed Said and Dr. Samir. they can very well understand that in our clinical practice, we see, and even oncology, that they're treating chemotherapy-induced anemia. I mean, in clinical practice, for example, today we have Eprex. Next week it's not available. Darbepoetin is available. What can we do? We switch them. We switch Eprex to Darbepoetin. How many times we have done? Many times. So is there any evidence of safe switchability between the dissimilar drugs? These are not the same drugs. These are different molecular structures. EPO alpha is different from EPO beta, and that's different from Darbepoetin, that's different from long-acting EPO. But it happens in the clinical practice. What about switching between the molecularly similar drugs? Eprex to its biosimilar. Is there any evidence of safe switchability? Yes, there is evidence of safe switchability. Yet the medical societies, their position statement is addressing only the switching between the molecularly similar drugs. So in order for a biosimilar to be harmful, one can think of only one mechanism, and that's immunogenicity. And in order for the biosimilar to have a carried over effect of immunogenicity, it has to be immunogenically a different molecule. Actually, I'm talking about the basic science because that's only I can address the concerns about switchability and extrapolation. My colleagues, physician colleagues, they will talk about the clinical trials and their incorporation is but this basic science, pharmaceutical science, is important to understand the concept of switchability, extrapolation, some other concepts. In order for a biosimilar to have a harmful, it should be immunogenically different molecule. And that's not possible because international regulators, they don't allow an immunogenically different molecule to be designated as a biosimilar. So in theory, this risk is very small. So guess how, there's a question, how many clinical studies have confirmed the safe switchability between the reference medicine to the biosimilar? As of April 18, 94 studies has demonstrated safe and successful switchability from the biological reference medicine to the biosimilars. One of the systematic review published in 2018 has provided assurance to the healthcare professional and public that this switchability is very safe. What is the risk of not switching? If you don't want to switch from the biological reference medicine to the biosimilar, is there any possibility that the patient is going to receive a different version of rituximab or trastuzumab or bevacizumab? Let's go through that. The answer is yes. You know, the biological medicines are complex protein structure. It's very difficult to maintain their molecular structure over time. It's fair to say the monoclonal antibodies like rituximab or trastuzumab, which were approved like more than 20 years ago, their structure right now, the version which we have right now, is not similar to, or is not identical. It's similar, but it's not identical to the version which was there at the time of launching or authorization, marketing authorization. So it's theoretically, it's fair to say the biological reference medicine itself, which is the originator, is a biosimilar of itself, because it's not the same version. International regulators, what they do, they ask these manufacturing companies that they have to do, if there's any manufacturing process change, if there's a variation in the version, then the international regulators demand these manufacturing companies to do pre and post manufacturing process change compatibility exercise in order to ensure the safety, efficacy, quality, immunogenicity of the new version is not adversely affected. Some of these biologicals, they have more than 50 versions as a result of the manufacturing process change, such as trastuzumab. And around 80% of the monoclonal antibodies, they have at least one manufacturing process change annually. And not switching to the biosimilar does not mean that your patient is not going to have a new version of the biological. Around 75% of these manufacturing process changes are carrying high or moderate potential risk. There are two ways of variation in the biological products. The way the biologicals are distinguished from the small molecules are their ability to have microheterogeneity. Microheterogeneity means they are vulnerable to the environmental factors like temperature. So variation in the temperature and environmental factor can result in alteration of the molecular structure, and that can alter the biological properties even of the biologicals. And the second source of the variation is the manufacturing process change. And one of the study had shown there has been at least 11, mean of 11 changes has happened to each biological. So this is what I told you, pre and post manufacturing process change compatibility exercise is mandatory requirement by international regulators in order for the new version to be released for the use in the patients. The biologicals are monoclonal antibodies, their structure is very complex. And one of the things which makes the structure more complex is the post-translational changes. One of the most notable post-translational changes is the glycosylation profile. This is the primary amino acid sequence, and these blue triangles are the glucose or sugar residues which are attached to the amino acids. So in order for this version to be immunogenically similar, you should have the same glycosylation profile. Even the primary amino acid sequence is the same, but the changes in the higher order structure can result in alteration of the biological properties, so that's not allowed. So this is happening in between the different versions or between the different batches of the biological, as well as when you are trying to demonstrate the biosimilarity, it should have the same glycosylation profile. So now I have proven and addressed the concern of immunogenicity. Theoretically, it's immunogenically similar molecule. So switchability is no issue at all. So we have addressed this concern. Now we can go to the extrapolation concern, the misconception about extrapolation. So the same concept of switchability, like there is different variations of the monoclonal antibodies. One of the medication called trastuzumab, one of the study had shown in 17 years, it had, you know, how many different versions? Guess how many versions it has? 17 years. It has 52 variants. So you have 52 variants of trastuzumab in 17 years. Now we are 2020, we're talking about 60 versions. Similarly, rituximab and others, there's more than 50 versions of these monoclonal antibodies. So extrapolation is not a new terminology. Extrapolation is a well-studied scientific principle, which is routinely used for a biological, which has several indications. For example, rituximab, it has several indications. With trastuzumab, it has several indications. Bevacizumab, it has several indications. It undergoes a major manufacturing process change. Then the international regulators, they mandate the manufacturer to do the pre and post compatibility exercise in one of the most sensitive indication as a clinical trial. Now I'm not talking about physical chemical characterization, I'm talking about clinical trial in one of the most sensitive indication. And you know, those who my physician colleagues and clinical pharmacists, they know that the sensitive indication is the one indication where they can show bigger magnitude of the response rates. So for example, for trastuzumab, early stage breast cancer is the most sensitive indication where you can show the PCR, pathological complete response rates, and you can easily demonstrate within 3 months or 2 months, Dr. Majid, like within 2 or 3 months, you can, because you have to give like 4 cycles, and then you look at the PCR and you can report it's 50%, 60%. You can easily do it. If it is compatible in that sensitive indication, used based on the totality of the evidence driven from this most sensitive indication, this data can be utilized to include all other indications of the biological reference medicine. Same concept applies for the biosimilars, and the same concept applies for the different versions or different batches of the same biological reference medicine. This is called extrapolation. So it's something which is happening even with the biological reference medicine and with the biosimilars. One of the examples was the subcutaneous trastuzumab. It was tested in one indication and it was extrapolated into all other indications. And the difference of this pharmaceutical difference of this magnitude is not acceptable for the biosimilars. Biosimilar, they have to be similar. This is subcutaneous, it has hyaluronidase and other stuff, which is not acceptable for the biosimilars. So other examples like filgrastim was approved for thrombocytopenia prevention, and it was extrapolated for stem cell mobilization. And EPOs, they were approved for preventing anemia with chronic kidney disease, and they were extrapolated for chemotherapy-induced anemia. The pharmacovigilance issues will be discussed in details by Dr. Iman, but you know there is not enough details about the efficacy, safety, and immunogenicity of biosimilars because it has been tested only in one clinical trial. So the effective pharmacovigilance is very important. Rigorous and effective pharmacovigilance is very important, and probably that's the reason we are here to understand what is pharmacovigilance. Dr. Iman will talk about it and implement it at Ministry of National Guard and Health Affairs. Because the WHO and international regulators, they mandate that the manufacturers, they should have a system in place which can detect, prevent, and report adverse drug-related events associated with the medications such as biosimilars and generics. This system is called pharmacovigilance. For that, actually in the hospital, we should have very good health information system, good communication system. In our hospital, we have ARIA. In ARIA, it's pretty much easily doable, but in best care, sometimes it's difficult. For example, when you're using filgrastim, you don't know this filgrastim is biosimilar versus this filgrastim is Neupogen. So we don't want to have this kind of thing, because if anything happens, so we should be able to associate the right event with the right product, like what was ordered versus what was received. And I think so that's pretty much doable with these blockbusters through the area. And naming strategy is very important. The US FDA has published a naming strategy. I think Dr. Iman will talk about it, but we have approved our own naming strategy for biosimilars, which is much more easy to understand and differentiate biosimilar from the reference medicine. So what we have recommended at Ministry of National Guard and Health Affairs, we have recommended to use the international non-proprietary names such as rituximab under bracket biosimilar hyphen, under brackets is brand name and hyphen will be biosimilar. So you'll be easily able to see this is biosimilar or this is the reference medicine. We published a paper a couple of years ago. My colleague, Dr. Hani, Dr. Asiri, Dr. Majid, Dr. Majid, so we published this paper and we mentioned this naming strategy in our paper. And that was highly appreciated by people around the Middle East. This was the paper published a couple of years ago. The biosimilars were approved first in Europe in 2006 when EPO and GCSF were approved, followed by a bunch of the other biosimilars. And the concept of introduction of the biosimilar in US was a bit late. First biosimilar was approved in 2015, followed by many other blockbusters biosimilars. Then Saudi FDA has initially approved filgrastim and epoetin, followed by recently trastuzumab, rituximab, and bevacizumab were approved by Saudi FDA. Here, I would like to mention a point. It's very important to know the country of origin for the biosimilar. What people around the world, the experts in the biosimilar around the world I met with, like Dr. Paul Cornish and many others from UK, from Canada, from the US, the biosimilars, the good biotechnology companies like Amgen and others, those who are located in US or Europe. These are the best biosimilar companies. So if we are accepting a biosimilar, this is what we have gone through. We have accepted the biosimilar mainly from US and the North America and the Europe, because they have good, reputable, not only good, reputable biotechnology skills, but also persistent and consistent supply chain. This was one of the landmark studies. I think Dr. Fouhedi will talk about it. So this was a LILAC study in which the biosimilar ABP 980 was compared with Herceptin trastuzumab in early stage breast cancer. And you can see the co-primary endpoint was the pathological complete response rates, and they were quite comparable. So if this study has shown the biosimilarity in the most sensitive indication, it can be extrapolated to include other indications as well. There is evidence of use of trastuzumab in international guidelines such as NCCN. These are trastuzumab-based combinations. And then there's a statement from FDA, from the NCCN, that FDA-approved biosimilar is an appropriate substitute for the trastuzumab, as well as in metastatic breast cancer. Now, some pharmacoeconomic aspects. So, Hani, how much time? 15 minutes, okay. So I think we have enough time. So let's go through the pharmacoeconomic perspective and the formulary consideration of the biosimilars in oncology. Why it is important to integrate biosimilars into the oncology practice. One of the studies had shown that global expenditures for the oncology is expected to reach like $66 billion a couple of years ago, which we have already passed through. So the only way we can reduce these expenditures if we can have this is for biologicals, by the way. The only way we can reduce these expenditures is if we can have the cost-effective, good quality, appropriate biosimilars, which can reduce the expenditures by 30 to 50%. And that amount of revenue can be used to expand the access of these cost-effective biologicals to treat more cancer patients around the world. And one of the study published in Future Oncology a couple of years ago, 3-4 years ago, had shown that if we switch 20,000 patients from Neupogen to biosimilar filgrastim Zerzio, which we have in our formulary, we can generate an amount of revenue which can be used to treat follicular lymphoma patients based on with obinutuzumab bendamustine for 60 to 169 patients, and also non-small cell lung cancer patients, around 21 to 60 patients, using pembrolizumab for the treatment of non-small cell lung cancer patients, which is very significant. This range is because of the 5 days GCSF versus 14 days GCSF. So if the duration of GCSF for prevention of brownthrombemia is 5 days, the amount generated is enough to treat 60 follicular lymphoma patients. If the duration of the filgrastim is 14 days, then the amount which is generated is enough to treat 170 follicular lymphoma patients based on very expensive treatment modality, obinutuzumab and bendamustine. So let's take an example of Saudi Arabia. I'll take an example of breast cancer based on the Global Observatory Cancer Report of Saudi Arabia. The breast cancer patients incidence was 3,600 in year 2018. And the 5-year prevalence was 12,000 patients. And around 25% of patients, Dr. Fohedi will agree with me, 25% of patients of breast cancer are HER2 positive breast cancer. So we're talking about around 3,000 patients are on trastuzumab or Herceptin based chemotherapy or target therapy. So if we use the price of the Saudi FDA, one patient is going to cost 125,000 reals per year using the subcutaneous trastuzumab. And in our paper with me and my colleague, we predicted a couple of years ago that if we can switch the subcutaneous trastuzumab to biosimilar trastuzumab, we can reduce the expenditures for the country from 375 million reals to 187 million reals, and we'll be spending something with GDP, which is the GDP of the country, times 0.8, which is very cost-effective strategy. And we had proven that we were right. I'll come to that. Now, being a member of the P&T committee members, I think many of us are sitting in the pharmacy and therapeutic committee, which consists of physician and pharmacy colleagues and other nursing colleagues and quality and logistic colleagues. So if we would like to approve biosimilars, we are exactly in line with the 2030 vision of the kingdom. What is that? That our nation is ambitious in what we want to achieve. We will apply efficiency and responsibility at all levels. So being a member of the Pharmacy and Therapeutic Committee, it's our responsibility. We shoulder this responsibility to make sure that we are approving cost-effective treatment options, which does not compromise the quality and care of the cancer patients, but we reduce expenditures in a cost-effective manner. So for that reason, after the loss of the patency of the biologicals, so being a member of the P&T committee, we take this initiative and we try to choose and evaluate a biosimilar which comes from a good biotechnology reputation. It has good, consistent supply chain, because sometimes you approve a biosimilar and you add it to the formulary because it's cheaper, but their source is India, is not good quality, are coming from China, or you have a biosimilar you approved and then you don't have a consistent supply. So the next month you run out of it, and then you have to bring another one, and then pharmacovigilance issues and stuff like that. So you have to pick and choose a biosimilar which comes of a good reputational biotechnology skills. I'm talking about country of origin, and then evaluate the clinical trials, see the safety and efficacy and stuff like that, and make sure that this is good for our patient, good for our country, good for our institutions. Naming strategy, policy, administrative policies and procedures. The National Guard Health Affairs have gone through all these processes. We have our own APP for biosimilars. We have our own policy. We have naming strategy. We are leader in the country. We are leader in the region. Our perception about the biosimilar is very clear at the level of the Ministry of National Guard Health Affairs. And we are definitely, I feel proud to be part of the National Guard, that we are a national guidance for the other hospitals how to adopt the biosimilars and stuff like that. One of the experts in biosimilar, his name is Dr. Paul Cornish. Maybe some of you have met with him 1 year or 2 years ago when he was around. He said in UK National Health Service, so they have a program like agreement with the manufacturers. They sign 1 to 2 years contract, And during this 1 to 2 year contract, NHS is now going to change the biosimilar, and the company has committed to provide the biosimilar in timely fashion. In case they are not able to provide, then they have to be penalized, and the penalty is to provide the originator. The cost difference, according to him, should be at least 50%. In Europe, like most of the biosimilars, they are providing like 30 to 50%. At National Guard Health Affairs, we have tried to make sure that the cost difference is at least 50%, 50 to 60% actually. So this is the expenditures of the National Guard Health Affairs. If Dr. Hani allows me, I can share with you. 5 years ago, trastuzumab expenditures was the most expensive oncology medication with 21 million reals. Rituximab, I'm mentioning because these are three medications I'm presenting today. Rituximab, 18 million, and bevacizumab, 11 million reals. So, Dr. Ahmed Saeed, Dr. Hani, you know, you're in higher administration. Every year we had been seeing like 10% increment in number of patients and as well as in the expenditures. So we extrapolated based on this, and in our paper we published this figure that we are expecting to spend 65 million reals by the year 2019, and Ministry of National Guard, which includes all hospitals of the National Guard. And we proposed, when we wrote this actually in the paper, many people from the industry, they came to me, how can you say that? We are Apple, we are Silicon Valley, we are San Francisco, nobody can break us. I said, when we will talk about, when we start negotiation of the biosimilar, you guys will reduce the price by 30%, and the biosimilars will reduce another 20 to 30%, and we'll eventually enjoy 50 to 60% cost saving benefit. I said, no way, it can never happen. And we made sure that it happened. This is what we predicted in our paper, and this is what we have done. So this is the formulary management. I'm just discussing with you the decision that we have taken. We have approved trastuzumab biosimilar. 420 milligram, Kanjinti is the brand. And expected expenditures was 27 millions. Now the expenditures are 11 million reals. So we are saving 16 million reals from this switch. And we're completely switching. Switchability and extrapolation is no more a concern. So that's what we have gone through in this lecture. And rituximab was a bit tricky because we have the second generation of the rituximab available, which is subcutaneous formulation of the rituximab. It's very difficult to completely switch rituximab because it saves infusion center a lot of time. Subcutaneous rituximab is given over 5 minutes. For example, imagine like you have an infusion center chair and you are giving rituximab. So this patient will occupy the whole day, a whole shift, using the same IV rituximab. But if you have subcutaneous rituximab, you can administer as much as like 10 patients or maybe even more. Mr. Umar, maybe you know better. Like using the same infusion center chair, you can administer as much as 10, maybe even more patients. So that has an hidden value, which is unbeatable. So you cannot replace subcutaneous rituximab in the indications, lymphoma, like where it has to be used. But there are indications, and this is resulting in, because also subcutaneous is cheaper, so this is resulting in 3 to 5 million reals. We did this shift like 2 years ago, but we implemented in 2020. You're gonna So this is the summary that I was telling you, example of the trastuzumab. We were able to save 16 million reals. We predicted in our paper that we'll be spending 11 million reals in 2019, but actually in 2020, which we were supposed to spend even more, we'll be spending 11 million reals. So this is what we have predicted, and this is what we have proven that we were right. Filgrastim, biosimilar filgrastim has already been used in National Guard Health Affairs since 2014. And when we talked about the biosimilar filgrastim, the company of the Neupogen, they reduced the price by 50% immediately right after the negotiation. This is what we were predicting. And after the 77% switch to Zerzio, we were saving 4.3 million real. And if we take into account the impact of this saving, we have saved 60 million reals up until 2019 by doing this switch, and our patients are doing fine. And I'll show you, and also there has been some other changes in the formulary. Neupogen is going to be deleted from the formulary completely, and we will have the biosimilar, and that's going to have around 10 million real saving per year now. And we are very unique hospital in the Middle East. We are the only hospital who has used filgrastim for the mobilization of the stem cell. Dr. Samir, you know it very well. You are part of this study. Dr. Samir is part of this, one of the resident project. We compared the biosimilar filgrastim versus the Neupogen for the mobilization of the stem cells. Nobody in the Middle East had this much great and brave heart to test this stem cell patient for the mobilization, but we had. Alhamdulillah, and we had proven it. They are similar. So the primary endpoint was the median CD34 stem cell collection. You can see in the both groups, in autologous stem cell transplant and in allogeneic stem cell transplant, there's no difference. There's no difference in the p-value. The amount of the stem cell collected were similar. The median time to neutrophil engraftment is similar. There's no difference. And the cost, which is associated with the use of the Zerzio, is 3 times cheaper. Last slide. There are some challenges which are faced by the first generation biosimilars, by the second generation, like rituximab, because they come with subcutaneous formulation. They provide convenient method of administration. They save infusions a lot of time. So it's very difficult to compete with them. And not only this, if you're doing pharmacoeconomic evaluation, you have to do cost minimization analysis if you're comparing two intervention A is equal to B, like first generation biological versus the first generation biosimilar. They are similar, so you don't have to do the cost effectiveness analysis. But if you're comparing with the first generation biosimilar versus the second generation biological reference medicine, then you have to do the cost effectiveness analysis. For example, the time of administration, how many patients you can use the infusion center to administer. So rituximab was very hard to completely switch, but trastuzumab is doable. With that, I would like to conclude that the biosimilars are very similar copy of the biological reference medicine. Switchability and extrapolation issue we have addressed today, and I'm sure everybody understands switchability is fine, extrapolation is fine, and I hope that we will be able to implement them successfully, inshallah, in the Ministry of National Guard Affairs. Thank you so much.
Thank you very much, Dr. Mansour. I know that it was challenging with time tight and too many slides to be shared. Now, if you allow me all, I know that there are some raised hands that the platform, so we can get the questions from there. So, Dr. Mateb, raise hand. So, Dr. Mateb, تفضل.
Thank you so much, Dr. Mansour Khan, for